Hi everyone, welcome, and it's my great pleasure to introduce you Páll-Gergely, who is a senior research fellow at the Plant Protection Institute in the Center for Agricultural Research. You should know about Barna that he spent a lot of time in Japan, 7 years. He made his PhD there and just returned 7 years ago to Hungary and take this position, and he loves snails.
Thank you. I think that was a very, very good introduction. Thank you very much. Ha nem hallotok, szóljatok, vagy ha valami, jó. So, I'm going to talk about land snail shells. They are beautiful. When I was 5 years old, I started to collect marine snail shells. They are bigger, more colorful, and so on. But I realized that land snail shells are also very, very interesting. And I have to admit that I'm a taxonomist, and this is the fate of taxonomists. So we discover Earth's biodiversity, we find wonderful new species in the nature, but Professor Barta invites me when I'm publishing an evolution paper, so I'm here to present something which is not strictly about taxonomy, but of course I will talk a little bit about species level diversity as well. For example, just very briefly, this is one of the recent works. It's 48 new species of a land snail genus from Southeast Asia. The name of the genus is Angustopila, and you can see here the scale. So practically all of them are 1 millimeter or smaller than 1 millimeter, and they live in caves and in rock crevices. And here there is a very nice map that a colleague made us. This one means that there is a single species there. I mean, in the soil sample that we collected. This larger and darker spot means that there are 2 to 3 species in the same locality. And there are some very nice sites with up to 7 species within the same soil sample. So actually, why I would like to show this kind of thing is that still it is possible not just to describe 1 or 2 new species from here and there, but to discover an entire, let's say, an entire branch, an entire group of organisms which was practically unknown in Southeast Asia. And with huge number of species. So this is the paper why I'm here. We submitted it to Evolution and it got accepted. He is Sipos András, or in English, András Sipos. He is the co-first author of the paper. So I will talk also a little bit about the story. So I'm looking at shells, I'm looking at specimens, and I had some interesting ideas about, well, interesting for me. I had some ideas about shells, and I contacted him. I found him quite by chance on the web. He works in the Budapest University of Technology and Economics. I think that's Műegyetem in Hungarian, yes. So entirely different branch of science, but he deals with shapes in the nature, like how a stone is formed. Why does a piece of stone look like that? Or how the root grows in the soil, these kind of topics. And I contacted him, and in the last 4 years we were working wonderfully on this project, and it's very, very interesting. I can suggest it to everyone. Try to find someone who does entirely different things than you and try some collaboration. And I also mentioned the other co-authors, mostly Thomas Neubauer. He's a friend of mine, a paleontologist. He made a lot of data entry for this paper, and I need to admit he's also much smarter than I am. So I'm just a snail guy. So what is this? Very, very, very good. Very, very good. This is an Etichiga land snail. It's, in English, it's called Roman snail or Helix pomatia. But now I have to stop for 1 second because a few years ago it turned out that this species, what we called Etichiga or Helix pomatia in whole Europe, from Greece to even to Germany, it's actually 2 species. And the Czech colleagues who wrote the paper, they said that they were from Romania, they were going back to Czech Republic, and they stopped, you know, for a double ladder pose in the Bükk Mountains in Hungary. And they found it in Hungary. So this is the Bükk Mountains, and we also tried to sample it as widely as possible, and here we have this species. Its name is Helix thessalica. The only difference is that its penis is more dark. So it has a more, but molecularly it's very distinct. And anyway, this is just a little addition that we know species, we know that there are common species, but always we can find something interesting thing. So going back to this one. So this is what we know about land snails. But actually, when we say land snails, it's not a monophyletic unit. For example, there is another type of land snail here. So what are the differences? For example, the eyes are here of this common European land snail at the end of the tentacle. But for other groups, it's mostly at the base of the tentacle. For example, that's only one difference. And another difference is this little door here. It's like a trap door. When the snail is disturbed or when the snail wants to rest, goes back to the shell, and this door seals the aperture completely. And this is a very nice defense mechanism against water loss and predators. Okay, now the second fun fact about snails. You know, I'm traveling to New Caledonia every year. This year I was there for the 5th time. It's a small oceanic island, and there is this genus Pararitida. So this snail belongs to this group. You see the tentacle, it looks like a normal snail, but it has a so-called pseudo-operculum. So it's a convergent evolutionary thing there. It's very, very remarkable. So the same thing happens, the snail goes back, and this thick part of its foot closes the aperture. I promise I will not keep going with this kind of fun facts about snails. So let's look at the numbers. So the group where our Roman snail belongs to, nem nagyon látszik. Don't you have a stick or something? Anyway, okay, it's maybe better. So this one, this group, Stylomatophora, it's not an easy word. This is a more difficult word anyway. So it has about 22,000 species worldwide. And the second group, Cyclophoroidea, it's a superfamily. It has 3,000 and something species. These don't live here in Europe. Oh, sorry. Some species live in Europe, but they are very rare. And there is this group. It's another group related to marine snails. This is a protected species, for example, in Europe. So what I was listing here, these are monophyletic groups that went from the water, mostly from marine environment, to terrestrial environment. And throughout the history of snails, this happened about 30 times. 30 times. And these are the 2 largest, and I'm going to talk about these 2. Okay, let's go a little bit further. Another little taxonomic thing is that in this presentation, every shell I'm showing is new species I described. So this is another place where I can put my new species. So here you can see some examples of the operculate snails. So these are the ones with the door, and these are the ones without the door, okay? What we can see is that the aperture is almost always rounded because the operculum is also rounded. So this is like an evolutionary constraint. And the aperture of these are mostly not rounded. I don't know if I can even find one example where the aperture is really rounded. And these have a lot of teeth inside in some groups. It can be very narrow. Probably it's good against predators. They cannot go so easily inside a shell. And you can think in another way that desiccation means water loss and protection against predators. These are the two most important things to be afraid of if you're a land snail. And for this group, for the first group, tényleg. Köszi. So for this group, it's already solved. And for this group, they have to be a little bit more innovative. So my idea was, okay, here we have a cross-section, a CT cross-section of the operculate group and the non-operculate group. So what do we see? We can see that if this one has to build the next wall, it touches at one point or at a very small part to the previous wall. You see? So this red part, which has to be built new, it's much bigger than here, right? So that was the original idea, that for this group, the shell is less economic than for this group, and this is because they always have the circular operculum, and this is an evolutionary constraint. But let's go back a little bit to see what other differences are between these two groups. So these ones are gonochoric. It means that there are boys and girls. They mostly live in the tropics. There are practically no invasive species and no shell reduction. Shell reduction means there are no slugs and semi-slugs in this group. But in the other groups, they are hermaphrodites, and you can think that if an animal is hermaphrodite, it can spread much easier, right? Because probably one specimen is enough to make a colony. They have a much wider distribution, not restricted to the tropics and subtropics. There are several invasive species. If you have a garden, you know that. And the shell reduction happened approximately 40 times. That's, again, a crazy topic. Shell is very good for snails. It provides shelter and protection, but throughout the evolution, at least 35, 40 times, it has been reduced. So we can say that this group is actually less successful and less innovative in evolutionary times than this group. I will talk about another difference in shell shape, but maybe we have to stop here for a few seconds to understand this graph. So these are from Arthur Kane's papers, 1977 and 78. So what he was doing is that, okay, this is the shell diameter and this is the shell height. So for example, let's say this is 3 millimeter times 1 millimeter, okay? It means that 1 millimeter width, 3 millimeter height. So the snail is somewhere there. I hope it's understandable. It's not so difficult to understand. And for this one, let's say it's 4 millimeter wide and let's say 1 or 2 millimeter height. So maybe it's this one. Of course, this is an Asian one and these are data from Europe. But from wherever the world you take the data, it looks like this for the Stylomatophoran land snails. And this one, this line means the width and the height is the same. And remarkably, there are very, very few snails there. Actually, the Etichiga, the land snail I was showing, it's actually somewhere there. So it has the same height and width, but that's absolutely very rare. So land snails are either flat or high-spired, tall. So this is the group with the door, okay? We can see some kind of similar pattern, but as you can see, there are much more examples on this line. So in this group, somehow, it's much more frequent proportionally to have snails with comparable height and width. And there was no explanation for that, and I think, or we think, that we have the answer, and, well, you need to believe me about this. So what about the evolution, I mean the chronological situation of these two groups? Because we can assume that the older group must be more diverse and more species rich and so on. And it's actually true. So the Stylomatophoran group, the one which lives here in Hungary, and the other one which has the door, they are different in age. Stylomatophoras are about 100 million years older, so much older group. But okay, this one, late Jurassic and early Jurassic, let's forget it, because this is based on 2 and 3 species worldwide. So it's so few data that we cannot say anything about this. But you can see that in the Cretaceous, early and late Cretaceous, the cyclophoradians, they gave, let's say, 1/3 of the species. And gradually it decreased until today. And again, let me show you some new species that I described. This is from the 99-million-year-old Burmese amber. And interestingly, in this very rich deposit, there are about 2,000 to 3,000 insect species described from this Burmese amber, and maybe about 40 land snails. And all land snails are the door ones, the ones with the door. And this one, Ferrucina petőfiana, it's named after Petőfi Sándor, and it's carrying the shell backwards. This is from the Hátszeg Basin in Transylvania. And this also belongs to this operculate group. So going back to this problem. So here is the part that I don't really like, because I'm very far from mathematics. But what András is saying, that we can think about an isoperimetric ratio, which is like this formula here. Szatmári Őrs always says that if you show one formula, then half of your audience will fall asleep. So please, I'm not going to explain it. What you need to understand is that if we are talking about a circle, then, okay, the perimeter is fixed. So in case of circle, the isoperimetric ratio is 1. But if the shape becomes more elongated, the isoperimetric ratio decreases. So it goes to, it approaches 0 in very elongated needle-like objects. And if we go one further, we will talk about a generalized isoperimetric ratio. So this green area is actually the area that we need to cover with a new shell material, okay? So this very nice equation here actually is talking about this perimeter here. And it can be larger than 1 because we are not talking about this one. This is already there. We only think about the outer one, which is marked as K2. And here is a very nice graph. You can imagine that these two circles touch each other, and we push the white circle into the green circle, okay? If we push it a little bit only, then it becomes a little bit higher, and if we push more, then it will reach a maximum at about 1.5. We are talking about circles. So in case of circles, the isoperimetric ratio will be maximum about at 1.5, and this is the most economic. So it means that we are using less material for this case. And there will be a decrease because we push it too much, and of course these shapes doesn't really, it makes no sense in the nature. But if we use ellipses, which is more common for the nature, for snails, the isoperimetric ratio can reach up to 3. So it can be even higher. And the larger isoperimetric ratio is the better. So what we thought is that it's not enough to do mathematics simulation and play with you know, circles that we draw, but we have to scan as many shells as possible. And this was the maximum that we could scan, 39 shells. And with this, we were trying to cover the morphological diversity of snails. And the methodology, very briefly, was that we got this row scan, and the software of András made this kind of whatever thing here. And I think, how many of this was? In the last 2 whorls, he was taking these kind of cross-section samples, and he was calculating the 2-dimensional isoperimetric ratio. And of course, we can calculate the volume and the surface. And we could use this data to understand what is going on with the snails. And here is one of the most interesting results that we get. So the green one is the snails with a door, with a circular operculum and aperture. And these ones are the ones without the door. So first of all, okay, what do we see here? Okay, the expanse angle. I will explain the expanse angle later. Sorry, it should be already here. But if the expanse angle is high, then the shell shapes are flat. If it's low, then the shell shapes are higher, like high-spired. And what you can see is that in high-spired snails, it doesn't really matter because the isoperimetric ratio is very high in both groups. But in flat groups, you can see that whatever you do, the green dots are close to the low values of isoperimetric ratio because they cannot really play with overlap. So it's a kind of fixed thing. And for the Stylomatophoraens, they can achieve quite high isoperimetric ratios with modifying the overlap between the whorls. So one way to increase the isoperimetric ratio is to make a bigger overlap. So this one is not very effective. Of course, poor snail cannot do anything because the shape is fixed due to the operculum, but the Stylomatophoraens, they can do this, but the operculus, they cannot do. So what can they do? They can increase the height. And if they increase the height, it will mean that one whorl doesn't, okay, here, this one meets with this one. But in case of higher spire, one whorl will meet with the previous one and the other one on the opposite side. So there will be a much bigger surface at which it doesn't need to make a whorl or doesn't need to make a strong whorl. So this is the only way that the operculate snails can make. And let's go back to this graph that I already showed you, because I'm going to show you very similar data in an entirely different graph. So here is this, what was the name of this? expansor angle, yes, thank you. This one is, okay, actually this is epsilon, and on the graph it will be some other Greek letters, sorry for that. But you see, when it's small, the shells are tall, and when it's big, the shells are flat, okay? So here, using our own data, we reproduce the old paper of Arthur Kane, and what can we see here? So this is the one with the door, okay? Here we have the relative frequency goes quite high in case of high-spired snails. It goes down, and in case of flat ones, it goes up again. So actually this one shows pretty much the same image. But as you can see, this valley here, it's much deeper for the other group. And I actually haven't mentioned, what do you think, why is it not good to have equal width and height? Do you have any idea why is it not good for a snail? It's a good question, right? Why is this gap there? Because these kind of shapes are quite unusual in nature. It's usually like a Gaussian distribution, but why is it not Gaussian? What do you think?
Easier to pick up if you have the same kind.
I will think about it, but currently this is not the explanation. So currently the explanation is about balance. So if it's flat, the snail carries it on the back. And if it's tall, the snail pulls it, okay? And if it's equal, I mean, if it's like a globular shape, it's very, very difficult to balance. And you can think of the helix pomatia. It's a snail with a very big aperture. It needs to have a very big aperture because it has a very big foot, a very big sole. It needs to have a lot of big muscles to keep that shell on the snail. And this is not very good if you have a good opening, because the predators can attack you very easily. So this is a kind of evolutionary trade-off, and snails like to avoid it. Okay, so for flat stilomatophorans, they can reach a high diversity with flat shape because they can play a lot with the whorl overlap. In the same time, flat operculate snails, of course there are many, but it's not working very well, and probably there is an evolutionary trade-off. So if they want to be more economic, they have to be more globular, which is difficult to balance. But still, here there is no such strong pressure because they can play with the whorl overlap. And yeah, this is the same thing I was talking about. So this globular shape is more economic for this group, but it's a little bit difficult probably to balance, but still proportionally there is a much higher number of species. And as I mentioned, for high-spired species it doesn't really matter because the isoperimetric ratio is the same. And I'm not an evolutionary biologist, but I think there is a term called adaptive valley, right? So you can think about this. The snail, why the flat snail doesn't evolve to have a tall snail? Because it starts evolving here to be more economic, but in the same time it's difficult to balance. And it cannot jump from here to there. There are examples, but in the whole, whatever, 20-something thousand land snails, there are very, very few examples where in flat groups there are high-spired members. So it's not adaptive. It cannot be adaptive. It's just by chance it happens. So, what I can say as a conclusion is that, of course, evolution of shell shape is made by evolution throughout various pressures, sexual selection, whatever, defense against specific predators. They have every kind of teeth, apertural barriers to avoid being eaten. But also, in the same time, it has to be economic. We are not saying that this is the most important and all snails want to save some material, but this factor certainly plays a role. And due to the biological constraints, which is the always circular operculum, this door snail group, the Cyclophoridae, they cannot be as variable in terms of shell shape as Stilomatophora, which don't have an operculum. And high spire shell shape is favorable in both groups because of the much higher isoperimetric ratio, but we cannot say that it is adaptive because there is an adaptive valley that the snails cannot jump. And that initial thing, what I mentioned, is that there are proportionally more globular snails in the door snail group. It can be due to this, that they want to achieve a more economic shell. And ultimately, of course, we cannot say that it's 100%, but the evolutionary success of the Stilomatophora group can be partly explained by this evolutionary constraint of the Cyclophoridae shell. So here I can stop. I would like to ask how many minutes do I have, because I have another little bit similar subject, about 10 minutes. Is it fine to go on with that? Okay. So I don't think I put this at the end, so think about this slide at the end. So this is one of the land snail groups I'm interested in as a taxonomist. But again, I'm not talking about species level diversity, but something very interesting. And I was talking about this in 2017 when I was here. I hope nobody remembers what I said then. So these are alisaids. It's one family. You see quite variable shell shapes, with strange operculum pattern and so on. And there is one crazy thing about this group, is this one. Okay, what do you think was that? Okay, I tried to help a little bit. So, okay, here is the aperture. So the snail comes out here, okay? That's clear. And this is the position of the door. That's the position of the operculum. So this thing is behind it. So anyway, historically, we know that many of these door snail groups, they have little snorkels, okay? In case of this shell and this shell, it's very obvious there is a little tube, okay? The snail doesn't have to come out to breathe, okay? But can stay inside, and gas exchange is perfectly going well and doesn't open the door, so nothing can attack it, and the water vapor doesn't go away, so everything is fine. And in some groups there are similar kind of slits, okay? So, okay, how can I go back with this one, right? Okay, let's go back a little bit. So this one was not very easy to think of because it's closed there. So what happens there? How can it work? And there was one observation, is that next to this little tube here, the shell surface is usually more ribbed. or if it's not more ribbed, then the rib density is a little bit different. So anyway, this region is always a little bit different. And there was this gentleman, maybe you know the K2 peak in the Himalaya. That's the most difficult to climb. And it was named after him. It was called Godwin-Austen Peak because he measured it first. So his job was to measure peaks in the Himalaya, and they made trigonometry, which means that he climbs one mountain and he has to wait until two other guys climb two other mountains, and he had a lot of time to collect snails. So he wrote that it should be noticed that the length of the tube in Aliceus bears invariably a constant relationship to the swollen, strongly ribbed portion of the body wall, and there is some intimate constructive connection between the two. So between the tube and that ribbed part. And the only other paper that deals with this is from 1964. Anyway, long text, long story short, this gentleman says that there must be microscopic holes on this tube, otherwise gas exchange wouldn't happen. So why is it there? But actually there are no such pores. If you look with scanning electron microscope, you find nothing. So actually by chance, I was observing this with scanning electron microscope, and I observed broken shells. And what I discovered is that there are microtunnels under the ribs. Incredible. Of course, he had no idea about it. Nobody had any ideas about it long time ago because there were no scanning electron microscope. So this is how it looks like. The one part of the shell is like folding over the other. And in plate tectonics, you know, there is the marine crust and the terrestrial crust. So I gave these names: anterior crust, posterior crust, and there is the microtunnel. So for this species, the diameter was like 16 micrometer. It's extremely narrow. And I was curious to find publications about how these very narrow tunnels work in physics or physical chemistry and whatever. No studies. It's so narrow that nobody makes it, nobody uses it, but why not? I mean, these happen in nature. Nobody thought that these kind of animals can have so complex structure. And here is a nano-CT image. So again, here you see these blue things are the microtunnels, and they go to the umbilicus. They open there, and they do the gas exchange to that tube. And the tube opens at a single hole inside the shell. And this is how the snail can breathe with entirely closed door. Not even a mite or a very tiny parasite can enter. So this is like a can, a conserve, and they can do this job. So anyway, we published this, we were happy with this, but since I knew what to look, where to look and what to search for, I found 3 other cases. There is this one where, okay, here you see it's a continuous tube, but in this case there are separated chambers, and each chamber is connected to 2 or 3 of these microtunnels. Okay, in this case, maybe it's a little bit difficult to understand, but you see here is the door. Here is the door, right? So this whole breathing apparatus is posterior to the door. Again, so door, and here is this breathing system. But for this group, the door is here, and this whole breathing system is anterior. It's in the other direction. I don't know if you can visually understand what is going on. And again, here is a tube, but here there is no tube. These individual little microtunnels, they go one by one to that single opening. And this is how the gas exchange works. And this is the fourth one. This is again an Asian group. Here, these microtunnels are very tiny. We cannot even indicate on this one, but they come out not in this direction, but in the upper sutural way. Anyway, I don't have other slides to explain it, but anyway, now we have these four systems. And we made molecular phylogeny because we were curious if this happened once or not. And it's very clear that during the evolution of these operculate snails, at least 4 times they thought that, oh, it would be good to make some kind of gas exchanging device. These three, this one, and this one are in Southeast Asia, and this one is from Madagascar. So initially we thought that these three must be somehow related, but no, it seems that this is the Aliseidae, this is Platyraphe, it's another operculate genus. This is Laotia, again here. It's very clearly separated from all other. And this one is Bucardicus from Madagascar. So 4 times they developed this kind of very, very precise, meticulous, and very nano breathing apparatus to avoid predation and desiccation. So yes, I put it there. Thank you very much for your attention.
Thank you. So it's very interesting talk. And
thank you for the invitation.
We'll have time for questions.
So when you're talking about economy of snail shells, do you mean synthesis costs or?
Yes, I mean, yes.
Do you know that building the shell is costly for a snail? Is there data?
Yes, that was also one question that I phrased at the beginning, but that time my Japanese supervisor said that in evolutionary biology everything is costly. So if you have a hair on your ear, that must have an explanation because that's costly. Well, I don't think everyone thinks like that, and that, of course I'm joking a bit. But yes, I think we can say that it requires energy, it requires material. Material is often not a problem, but in limestone rock areas there is unlimited source of calcium carbonate. But there are places where there is no such thing, and still snails live there. They need to find the material, they need to build it in their shell. So yes, it's a cost.
So the reason I'm asking is because if you know what the cost is, then you could build hypotheses to further explain this phenomenon, because if it's energy, of course energy or food availability could vary randomly across the environment, but if it's carbonate, which is the limiting factor, then you have already a very interesting question. Do you see the same constraint on a species that live on limestone area because they have unlimited access, and you would see a less deep valley, I guess, in between?
Yes, that can be a good point, and we were thinking about this. So we cannot. Okay, so for example, if you go to Southeast Asia, you visit the forest patch, you will find this members of the two groups, the door one and the one without a door, same size and so on. And if we measure a lot, we could say, we predict that the shell thickness and the weight of the two species should be different, because the one with the door, it needs to build a stronger shell, stronger outer wall to compensate this problem. And if we make enough replicates, maybe we can statistically prove that that this is really a meaningful thing for snails. But for that, you need to go to Southeast Asia and so on. But anyway, it's a good point. We thought, so your question, yes, it's a very good question. We didn't really bother very much about it. We thought that it's a default state, okay? Making shell is cost, and I don't know, what would you think with your evolutionary biologist brain? Do you think it's something to really consider and think about? Maybe this is, after all, not a very limiting thing.
Yeah, so it's definitely costly, but probably it can be a question of how costly.
Yeah, how costly.
So it might be, it still have a cost, but it's not a significant cost. And then that's the argument still.
Yeah.
And maybe a related question. You were mentioning balance as an important factor. So I would expect that for a small snail, since it's gravitation, but gravitation affects small size less than
that's true.
large ones.
That's true.
Do you see a difference as a function of size? So for small ones, you should see a smaller constraint on shell evolution.
That's true to a certain degree. So there are shell shapes which are carried like this by the snail if it's like a 2 millimeter species, and we know that if it would be like a 2 centimeter snail, it would pull it. So this is actually a good point, and we know several species which are actually flat shell when they are small. And when they grow, they become high-spired. I don't know if you can imagine. So the shell looks like this, okay? It means that the top is the embryonic whorls, and when the snail is a child, it's a flat shape. So the snail carries it. And it grows like this, and it becomes like a high-spire. And there is one point in the snail's life when the snail doesn't carry it but pulls it. And this is something what I mentioned to my friends, that this is a very interesting scientific question. when is that point when the snail starts to pull its shell instead of carrying? It's a good scientific question, but you cannot just explain it outside of these kind of rooms.
According to my high school biology teacher, a good scientist is a person who can talk a lot about small things.
Good. I like it.
Other questions? Yeah.
Following up on Yacin's questions, I had a similar question regarding economy costs. But in the context of sexual selection, so what about species where you have separate males and females? Can you see a difference in the shell structure? Because I would expect that if balancing is difficult, then maybe as a handicap, maybe males are carrying more difficult to balance shells than females. Do you see any evidence for this?
So far, there are very, very few reports of different shell shape, and in practically all but one example, it's only statistical. So it means that this one, the nyugati ajtós csiga, it's called Pomácias elegans. That's a protected species. It lives, for example, in Tihany in Hungary. You go there, you pick up 2 specimens. You will not tell if it's a boy or a girl. But if you make morphometrics, you will see that there are 2 highly overlapping clusters, but there are extremes which are very probably sexed. So you can say if it's too wide to be a male and too narrow to be a female. And actually, we found one case in central Vietnam where the girls were much larger than the boys. But so this is probably the only case when this happens. And in this group somehow there is no big how do you call it? Yes, thank you. No, somehow there's no big dimorphism, but bisexual selection, I didn't mean exactly that. So like mate recognition, okay, like the hairs or shell surface sculpture. It's in some groups it is said to be species specific, and this is a tool for the mating partner to recognize the snail. So rather this kind of thing.
And if I can have a related question, you mentioned that there are these two groups, and so in one group the hermaphroditism is much more common than in...
It's 100%.
Yeah, 100%.
Yeah, so in one group they are all hermaphrodites, and the other group they are all, I forget the word, I just looked it up in Wikipedia. So what is it?
Gonochoristic.
Yeah, gonochoristic. Thank you.
Yeah, so maybe I missed this, but can you remind me that what is the explanation for that? So why
Well, it's just different evolutionary history.
Okay.
So it's not adaptational, it just happened like that.
And one more question, if there is no more program audience. That all of your argument is based on the assumption that these dog snails are constrained by the shape of the door.
Yes.
It must be a circle.
Yes.
Why is this? What prevents these snails to develop an oval or a more complicated?
That's a very good question, because their relatives, which are marine and freshwater snails, they have all kind of operculum shapes, and sometimes it's even lost. I cannot answer, but as far as I understand the way of thinking of evolutionary biologists, you cannot really ask this question, right? I mean, so why one group can do something what the other cannot do? Or
say your argument is that there is quite a strong selection pressure.
Yes.
For having
more overlap, for example. Yes.
And
Yes.
different, so not a circle-shaped shell.
Why not? Why doesn't it?
And what prevent? So
Yeah.
and as you said, so there are examples that this can be done.
In other groups, in other clades, yes.
So
Yeah. Somehow in this clade, yeah, very good question, but yeah.
can it be the case that this is a kind of sexual selection case? Because Al-Zahabi has a quite famous examples that if you try to persuade someone that you are in a perfect shape, then you should produce a very difficult shape.
Yeah.
Produce a circle.
Can be a sign of perfection.
Yeah,
Yeah.
it's just a
Well,
idea.
yeah, but for this we would know how they make the courtship and so on, but we practically don't know. But yeah, why not? Why not? Yeah, thank you.
Other questions? Thank you. Then we are going to have a lunch in the mensa, so if you have still questions or just have a chat with Barna, then please join us. And,
Okay.
thank you very much.
Thank you very much.